The photo on the left shows large, stalk-like saguaro cacti with multiple arms, and the photo on the right shows a lizard on a rock. Figure 18. 1. All organisms are products of evolution adapted to their environment. A saguaro, Carnegie gigantea, can soak up 750 liters of water in a single rainstorm, enabling these cacti to survive the dry conditions of the Sonora Desert in Mexico and the southwestern United States. B. The Andean semi-aquatic lizard, Potomites montanicola, discovered in Peru in 2010, lives between 1,570 to 2,100 meters in elevation, and, unlike most lizards, is nocturnal and swims. Scientists still do not know how these cold-blooded animals are able to move in the cold, 10 to 15 degrees C, temperatures of the Andean night. Credit a modification of work by Gentry George, U.S. Fish and Wildlife Service. Credit B. Modification of work by Germán Chavez and Diego Vázquez, Zuquis. Chapter outline. 18.1 Understanding evolution. 18.2 Formation of new species. 18.3 Reconnection and speciation rates. All living organisms, from bacteria to baboons to blueberries, evolved at some point from a different species. Although it may seem that living things today stay much the same, that is not the case. Evolution is an ongoing process. The theory of evolution is the unifying theory of biology, meaning it is the framework within which biologists ask questions about the living world. Its power is that it provides direction for predictions about living things that are borne out in ongoing experiments. The Ukrainian-born American geneticist Theodosius Dobzhansky famously wrote that nothing makes sense in biology except in the light of evolution. He meant that the tenet that all life has evolved and diversified from a common ancestor is the foundation from which we approach all questions in biology. Learning objectives. By the end of this section, you will be able to do the following. Describe how scientists develop the present-day theory of evolution. Define adaptation. Explain convergent and divergent evolution. Describe homologous and vestigial structures. Discuss misconceptions about the theory of evolution. Evolution by natural selection describes a mechanism for how species change over time. Scientists, philosophers, researchers, and others had made suggestions and debated this topic well before Darwin began to explore this idea. Classical Greek philosopher Plato emphasized in his writings that species were static and unchanging, yet there were also ancient Greeks who expressed evolutionary ideas. In the 18th century, naturalist Georges Louis Leclerc Comte de Buffon reintroduced ideas about the evolution of animals and observed that various geographic regions have different plant and animal populations, even when the environments are similar. Some at this time also accepted that there were extinct species. Also during the 18th century, James Hutton, a Scottish geologist and naturalist, proposed that geological change occurred gradually by accumulating small changes from processes operating like they are today over long periods of time. This contrasted with the predominant view that the planet's geology was a consequence of catastrophic events occurring during a relatively brief past. 19th century geologist Charles Lyell popularized Hutton's view. A friend to Darwin. Lyell's ideas were influential on Darwin's thinking. Lyell's notion of the greater age of Earth gave more time for gradual change in species, and the process of change provided an analogy for this change. In the early 19th century, Jean-Baptiste Lamarck published a book that detailed a mechanism for evolutionary change. We now refer to this mechanism as an inheritance of acquired characteristics, by which the environment causes modifications in an individual or offspring could use or disuse of a structure during its lifetime, and thus bring about change in a species. While many discredited this mechanism for evolutionary change, Lamarck's ideas were an important influence on evolutionary thought. Charles Darwin and natural selection. In the mid-19th century, two naturalists, Charles Darwin and Alfred Russel Wallace, independently conceived and described the actual mechanism for evolution. Importantly, each naturalist spent time exploring the natural world on expeditions to the tropics. From 1831 to 1836, Darwin traveled around the world on HMS Beagle, including stops in South America, Australia, and the southern tip of Africa. Wallace traveled to Brazil to collect insects in the Amazon rainforest from 1848 to 1852 and to the Malay archipelago from 1854 to 1862. Darwin's journey, like Wallace's later journeys to the Malay archipelago, included stops at several island chains, the last being the Galapagos Islands west of Ecuador. On these islands, Darwin observed species of organisms on different islands that were clearly similar, yet had distinct differences. For example, the ground finches inhabiting the Galapagos Islands comprised several species with a unique beak shape, figure 18. 2. The species on the islands had a graded series of beak sizes and shapes with very small differences between the most similar. He observed that these finches closely resembled another finch species on the South American mainland. Darwin imagined that the island species might be species modified from one of the original mainland species. Upon further study, he realized that each finch's varied beaks helped the birds acquire a specific type of food. For example, seed-eating finches had stronger, thicker beaks for breaking seeds, and insect-eating finches had spear-like beaks for stabbing their prey. Illustration shows 4 different species of finch from the Galapagos Islands. Beak shape ranges from broad and thick to narrow and thin. Figure 18. 2. Darwin observed that beak shape varies among finch species. He postulated that ancestral species' beaks had adapted over time to equip the finches to acquire different food sources. Wallace and Darwin both observed similar patterns in other organisms, and they independently developed the same explanation for how and why such changes could take place. Darwin called this mechanism natural selection. Natural selection, or survival of the fittest, is the more prolific reproduction of individuals with favorable traits that survive environmental change because of those traits. This leads to evolutionary change. For example, Darwin observed a population of giant tortoises in the Galapagos archipelago to have longer necks than those that lived on other islands with dry lowlands. These tortoises were selected because they could reach more leaves and access more food than those with short necks. In times of drought when fewer leaves would be available, those that could reach more leaves had a better chance to eat and survive than those that couldn't reach the food source. Consequently, long-necked tortoises would be more likely to be reproductively successful and pass the long-necked trait to their offspring. Over time, only long-necked tortoises would be present in the population. Natural selection, Darwin argued, was an inevitable outcome of 3 principles that operated in nature. First, most characteristics of organisms are inherited or passed from parent to offspring. Although no one, including Darwin and Wallace, knew how this happened at the time, it was a common understanding. Second, more offspring are produced than are able to survive, so resources for survival and reproduction are limited. The capacity for reproduction in all organisms outstrips the availability of resources to support their numbers. Thus, there is competition for those resources in each generation. Both Darwin and Wallace's understanding of this principle came from reading economist Thomas Malthus' essay that explained this principle in relation to human populations. Third, offspring vary among each other in regard to their characteristics, and those variations are inherited. Darwin and Wallace reasoned that offspring with inherited characteristics, which allow them to best compete for limited resources, will survive and have more offspring than those individuals with variations that are less able to compete. Because characteristics are inherited, these traits will be better represented in the next generation. This will lead to change in populations over generations in a process that Darwin called descent with modification. Ultimately, natural selection leads to greater adaptation of the population to its local environment. It is the only mechanism known for adaptive evolution. In 1858, Darwin and Wallace, figure 18. 3, presented papers at the Linnean Society in London that discussed the idea of natural selection. The following year, Darwin's book, On the Origin of Species, was published. His book outlined in considerable detail his arguments for evolution by natural selection. Images of Charles Darwin and Alfred Wallace are shown. Figure 18. 3 both, A. Charles Darwin and B. Alfred Wallace wrote scientific papers on natural selection that they presented together at the Linnean Society in 1858. Studies of evolution by natural selection are difficult to conduct. as they require analyses of several generations of the investigated species in order to document the changes pointing to evolutionary change. The Galapagos finches are an excellent example. Peter and Rosemary Grant and their colleagues have studied Galapagos finch populations every year since 1976 and have provided important evidence of natural selection. The Grants found changes from one generation to the next in beak shape distribution, with the medium ground finch on the Galapagos island of Daphne Major. The birds have inherited a variation in their bill shape, with some having wide, deep bills and others having thinner bills. During a period in which rainfall was higher than normal because of an El Niño, there was a lack of large, hard seeds of which the large-billed birds ate. However, there was an abundance of the small, soft seeds which the small-billed birds ate. Therefore, the small-billed birds were able to survive and reproduce. In the years following this El Niño, the Grants measured beak sizes in the population and found that the average bill size was smaller. Since bill size is an inherited trait, parents with smaller bills had more offspring, and the bill evolved into a much smaller size. As conditions improved in 1987 and larger seeds became more available, the trend towards smaller average bill size ceased. Career connection. Field biologist. Many people hike, explore caves, scuba dive, or climb mountains for recreation. People often participate in these activities hoping to see wildlife. Experiencing the outdoors can be incredibly enjoyable and invigorating. What if your job entailed working in the wilderness? Field biologists, by definition, work outdoors in the field. The term field in this case refers to any location outdoors, even underwater. A field biologist typically focuses research on a certain species, group of organisms, or a single habitat, figure 18.4. Photo shows a scientist next to a tranquilized polar bear laying on the snow. Figure 18. 4. A field biologist tranquilizes a polar bear for study. Credit. Karen Road. One objective of many field biologists includes discovering new, unrecorded species. Not only do such findings expand our understanding of the natural world, but they also lead to important innovations in fields such as medicine and agriculture. Plant and microbial species, in particular, can reveal new medicinal and nutritive knowledge. Other organisms can play key roles in ecosystems or, if rare, require protection. When discovered, researchers can use these important species as evidence for environmental regulations and laws. Processes and patterns of evolution. Natural selection can only take place if there is variation or differences among individuals in a population. Importantly, these differences must have some genetic basis. Otherwise, the selection will not lead to change in the next generation. This is critical because non-genetic reasons can cause variation among individuals, such as an individual's height, because of better nutrition rather than different genes. Genetic diversity in a population comes from two main mechanisms: mutation and sexual reproduction. Mutation, a change in DNA, is the ultimate source of new alleles or new genetic variation in any population. The genetic changes that mutation causes can have one of three outcomes on the phenotype. A mutation affects the organism's phenotype in a way that gives it reduced fitness, lower likelihood of survival, or fewer offspring. A mutation may produce a phenotype with a beneficial effect on fitness. Many mutations will also have no effect on the phenotype's fitness. We call these neutral mutations. Mutations may also have a whole range of effect sizes on the organism's fitness that expresses them in their phenotype, from a small effect to a great effect. Sexual reproduction also leads to genetic diversity. When two parents reproduce, unique combinations of alleles assemble to produce the unique genotypes and thus phenotypes in each offspring. We call a heritable trait that helps an organism's survival and reproduction in its present environment an adaptation. Scientists describe groups of organisms adapting to their environment when a genetic variation occurs over time that increases or maintains the population's fit to its environment. A platypus's webbed feet are an adaptation for swimming. A snow leopard's thick fur is an adaptation for living in the cold. A cheetah's fast speed is an adaptation for catching prey. Whether or not a trait is favorable depends on the current environmental conditions. The same traits are not always selected because environmental conditions can change. For example, consider a plant species that grew in a moist climate and did not need to conserve water. Large leaves were selected because they allowed the plant to obtain more energy from the sun. Large leaves require more water to maintain than small leaves, and the moist environment provided favorable conditions to support large leaves. After thousands of years, the climate changed, and the area no longer had excess water. The direction of natural selection shifted so that plants with small leaves were selected, because those populations were able to conserve water to survive the new environmental conditions. The evolution of species has resulted in enormous variation in form and function. Sometimes, evolution gives rise to groups of organisms that become tremendously different from each other. We call two species that evolve in diverse directions from a common point divergent evolution. We can see such divergent evolution in the forms of the reproductive organs of flowering plants, which share the same basic anatomies. However, they can look very different as a result of selection in different physical environments and adaptation to different kinds of pollinators. Figure 18.5. Photo showing a dense blazing star, Liatris spicata, and a purple coneflower, Echinacea purpurea. The dense blazing star flower has purple strand-like petals budding from its stem. The coneflower has a large circular center of the budding flower, with purple petals growing from it. Figure 18. 5 flowering plants evolved from a common ancestor. Notice that the dense blazing star, Liatris spicata, and the B purple coneflower Echinacea purpurea vary in appearance, yet both share a similar basic morphology. Credit A, modification of work by Drew Avery. Credit B, modification of work by Corey Zanker. In other cases, similar phenotypes evolve independently in distantly related species. For example, flight has evolved in both bats and insects, and they both have structures we refer to as wings, which are adaptations to flight. However, bat and insect wings have evolved from very different original structures. We call this phenomenon convergent evolution, where similar traits evolve independently in species that do not share a recent common ancestry. The trait in the two species came to be similar in structure and have the same function, flying, but did so separately from each other. These physical changes occur over enormous time spans and help explain how evolution occurs. Natural selection acts on individual organisms, which can then shape an entire species. Although natural selection may work in a single generation on an individual, it can take thousands or even millions of years for an entire species genotype to evolve. It is over these large time spans that life on Earth has changed and continues to change. Evidence of evolution. The evidence for evolution is compelling and extensive. Looking at every level of organization in living systems, biologists see the signature of past and present evolution. Darwin dedicated a large portion of his book, On the Origin of Species, to identifying patterns in nature that were consistent with evolution, and since Darwin, our understanding has become clearer and broader. Fossils. Fossils provide solid evidence that organisms from the past are not the same as those today, and fossils show the gradual evolutionary changes over time. Scientists determine the age of fossils and categorize them from all over the world to determine when the organisms lived relative to each other. The resulting fossil record tells the story of the past and shows the evolution of form over millions of years, figure 18.6. For example, scientists have recovered highly detailed records showing the evolution of humans and horses, figure 18.6. The whale flipper shares a similar morphology to bird and mammal appendages, figure 18.7, indicating that these species share a common ancestor. Photo A shows a museum display of hominid skulls that vary in size and shape. Illustration B shows 5 extinct species related and similar in appearance to the modern horse. The species vary in size from that of a modern horse to that of a medium-sized dog. Figure 18.6. In this, a display, fossil hominids are arranged from oldest bottom to newest top. As hominids evolved, the skull's shape changed. An artist's rendition of B, extinct species of the genus Equus, reveals that these ancient species resembled the modern horse, Equus ferus, but varied in size. Anatomy and embryology. Another type of evidence for evolution is the presence of structures in organisms that share the same basic form. For example, the bones in human, dog, bird, and whale appendages all share the same overall construction, figure 18.7, resulting from their origin in a common ancestor's appendages. Over time, evolution led to changes in the bones' shapes and sizes in different species, but they have maintained the same overall layout. Scientists call these synonymous parts homologous structures. Illustration compares a human arm, dog and bird legs, and a whale flipper. All appendages have the same bones, but the size and shape of these bones vary. Figure 18.7, the similar construction of these appendages indicates that these organisms share a common ancestor. Some structures exist in organisms that have no apparent function at all and appear to be residual parts from a past common ancestor. We call these unused structures without function vestigial structures. Other examples of vestigial structures are wings on flightless birds, leaves on some cacti, and hind leg bones in whales. Not all similarities represent homologous structures. As explained in determining evolutionary relationships, when similar characteristics occur because of environmental constraints and not due to a close evolutionary relationship, it is an analogy or homoplasy. For example, insects use wings to fly like bats and birds, but the wing structure and embryonic origin are completely different. These are analogous structures, figure 20.8. On the other side, the bird and bat wings are homologous because the bones are inherited from a common ancestor, while the wings themselves are analogous as they evolved independently. Link to learning. Watch this video exploring the bones in the human body. Another piece of evidence of evolution is the convergence of form in organisms that share similar environments. For example, species of unrelated animals, such as the Arctic fox and ptarmigan, living in the Arctic region have been selected for seasonal white phenotypes during winter to blend with the snow and ice, figure 18.8. These similarities occur not because of common ancestry, but because of similar selection pressures, the benefits of predators not seeing them. The left photo depicts an Arctic fox with white fur sleeping on white snow, and the right photo shows a ptarmigan bird with white plumage standing on white snow. Figure 18.8, the white winter coat of the Arctic fox and the B, ptarmigan's plumage are adaptations to their environments. Credit a modification of work by Keith Morehouse. Embryology, the study of the anatomy of an organism's development to its adult form, also provides evidence of relatedness between now widely divergent groups of organisms. Mutational tweaking in the embryo can have such magnified consequences in the adult that embryo formation tends to be conserved. As a result, structures that are absent in some groups often appear in their embryonic forms and disappear when they reach the adult or juvenile form. For example, all vertebrate embryos, including humans, exhibit gill slits and tails at some point in their early development. These disappear in the adults of terrestrial groups, but adult forms of aquatic groups, such as fish and some amphibians, maintain them. Great ape embryos, including humans, have a tail structure during their development that they lose when they are born. Biogeography, the geographic distribution of organisms on the planet, follows patterns that we can explain best by evolution in conjunction with tectonic plate movement over geological time. Broad groups that evolved before the supercontinent Pangaea broke up, about 200 million years ago, are distributed worldwide. Groups that evolved since the breakup appear uniquely in regions of the planet, such as the unique flora and fauna of northern continents that formed from the supercontinent Laurasia and of the southern continents that formed from the supercontinent Gondwana. The presence of members of the plant family Proteaceae in Australia, southern Africa, and South America indicates that their ancestors were predominant on the supercontinent Gondwana prior to its breaking up. Marsupial diversification in Australia and the absence of other mammals reflect Australia's long isolation. Australia has an abundance of endemic species found nowhere else, which is typical of islands whose isolation by expanses of water prevents species from migrating. Over time, these species diverge evolutionarily into new species that look very different from their ancestors that may exist on the mainland. Australia's marsupials, the Galapagos finches, and many species on the Hawaiian Islands are all unique to their one point of origin. Yet they display distant relationships to ancestral species on mainlands. Molecular biology. Like anatomical structures, the molecular structures of life reflect descent with modification. DNA's universality reflects evidence of a common ancestor for all of life. Fundamental divisions in life between the genetic code, DNA replication, and expression are reflected in major structural differences in otherwise conservative structures, such as ribosome components and membrane structures. In general, the relatedness of groups of organisms is reflected in the similarity of their DNA sequences, exactly the pattern that we would expect from descent and diversification from a common ancestor. DNA sequences have also shed light on some of the mechanisms of evolution. For example, it is clear that the evolution of new functions for proteins commonly occurs after gene duplication events that allow freely modifying one copy by mutation, selection, or drift, changes in a population's gene pool resulting from chance, while the second copy continues to produce a functional protein. Misconceptions of evolution. Although the theory of evolution generated some controversy when Darwin first proposed it, biologists almost universally accepted it, particularly younger biologists, within 20 years after publication of On the Origin of Species. Nevertheless, the theory of evolution is a difficult concept and misconceptions about how it works abound. Link to learning. This site addresses some of the main misconceptions associated with the theory of evolution. Evolution is just a theory. Critics of the theory of evolution dismiss its importance by purposefully confounding the everyday usage of the word theory with the way scientists use the word. In science, we understand a theory to be a body of thoroughly tested and verified explanations for a set of observations of the natural world. Scientists have a theory of the atom, a theory of gravity, and the theory of relativity, each which describes understood facts about the world. In the same way, the theory of evolution describes facts about the living world. As such, a theory in science has survived significant efforts to discredit it by scientists. In contrast, a theory in common vernacular is a word meaning a guess or suggested explanation. This meaning is more akin to. It is also important to understand that the variation that natural selection works on is already in a population and does not arise in response to an environmental change. For example, applying antibiotics to a population of bacteria will, over time, select a population of bacteria that are resistant to antibiotics. The resistance, which a gene causes, did not arise by mutation because of applying the antibiotic. The gene for resistance was already present in the bacteria's gene pool, likely at a low frequency. The antibiotic, which kills the bacterial cells without the resistance gene, strongly selects individuals that are resistant, since these would be the only ones that survived and divided. Experiments have demonstrated that mutations for antibiotic resistance do not arise as a result of antibiotic application. In a larger sense, evolution is not goal-directed. Species do not become better over time. They simply track their changing environment with adaptations that maximize their reproduction in a particular environment at a particular time. Evolution has no goal of making faster, bigger, more complex, or even smarter species, despite the commonness of this kind of language in popular discourse. What characteristics evolve in a species are a function of the variation present and the environment, both of which are constantly changing in a non-directional way. A trait that fits in one environment at one time may well be fatal at some point in the future. This holds equally well for insect and human species. Learning objectives. By the end of this section, you will be able to do the following. Define species and describe how scientists identify species as different. Describe genetic variables that lead to speciation. Identify prezygotic and postzygotic reproductive barriers. Explain allopatric and sympatric speciation. Describe adaptive radiation. Although all life on Earth shares various genetic similarities, only certain organisms combine genetic information by sexual reproduction and have offspring that can then successfully reproduce. Scientists call such organisms members of the same biological species. Species and the ability to reproduce. A species is a group of individual organisms that interbreed and produce fertile, viable offspring. According to this definition, one species is distinguished from another when, in nature, it is not possible for matings between individuals from each species to produce fertile offspring. Members of the same species share both external and internal characteristics, which develop from their DNA. The closer relationship two organisms share, the more DNA they have in common, just like people and their families. People's DNA is likely to be more like their father or mother's DNA than their cousin or grandparents' DNA. Organisms of the same species have the highest level of DNA alignment, and therefore share characteristics and behaviors that lead to successful reproduction. Species appearance can be misleading in suggesting an ability or inability to mate. For example, even though domestic dogs, Canis lupus familiaris, display phenotypic differences, such as size, build, and coat, most dogs can interbreed and produce viable puppies that can mature and sexually reproduce. Figure 18. 9. Photo A shows a poodle with curly short fur. Photo B shows a cocker spaniel with long, wavy fur that has light brown parts and cream-colored markings on the face, forepaws, belly, hind legs, and tail. The poodle has longer legs than the cocker spaniel. The cockapoo in Photo C has curly hair, like the poodle, and short legs, like the cocker spaniel. Figure 18. 9. The A poodle and B cocker spaniel can reproduce to produce a breed known as C. the cockapoo. Credit A modification of work by Sally Eller, Tom Rees. Credit B modification of work by Jeremy McWilliams. Credit C modification of work by Kathleen Conklin. In other cases, individuals may appear similar although they are not members of the same species. For example, even though bald eagles, Haliaeetus leucosphalus, and African fish eagles, Haliaeetus vocifer, are both birds and eagles, each belongs to a separate species group, figure 18. 10. If humans were to artificially intervene and fertilize a bald eagle's egg with an African fish eagle's sperm, and a chick did hatch, that offspring, called a hybrid, a cross between two species, would probably be infertile, unable to successfully reproduce after it reached maturity. Different species may have different genes that are active in development. Therefore, it may not be possible to develop a viable offspring with two different sets of directions. Thus, even though hybridization may take place, the two species still remain separate. Photo A shows a picture of the African fish eagle in flight, and Photo B shows the bald eagle perched on a post. Both birds have dark brown feathers on their bodies and wings, and white feathered heads. Figure 18. 10. The A African fish eagle is similar in appearance to the B bald eagle, but the two birds are members of different species. Credit A modification of work by Nigel Wedge. Credit B modification of work by U.S. Fish and Wildlife Service. Populations of species share a gene pool, a collection of all the gene variants in the species. Again, the basis to any changes in a group or population of organisms must be genetic. For this is the only way to share and pass on traits. When variations occur within a species, they can only pass to the next generation along two main pathways: asexual reproduction or sexual reproduction. The change will pass on asexually simply if the reproducing cell possesses the changed trait. For the changed trait to pass on by sexual reproduction, a gamete, such as a sperm or egg cell, must possess the changed trait. In other words, sexually reproducing organisms can experience several genetic changes in their body cells. But if these changes do not occur in a sperm or egg cell, the changed trait will never reach the next generation. Only heritable traits can evolve. Therefore, reproduction plays a paramount role for genetic change to take root in a population or species. In short, organisms must be able to reproduce with each other to pass new traits to offspring. Speciation. The biological definition of species, which works for sexually reproducing organisms, is a group of actual or potential interbreeding individuals. There are exceptions to this rule. Many species are similar enough that hybrid offspring are possible and may often occur in nature. But for the majority of species, this rule generally holds. The presence in nature of hybrids between similar species suggests that they may have descended from a single interbreeding species, and the speciation process may not yet be completed. Given the extraordinary diversity of life on the planet, there must be mechanisms for speciation. the formation of two species from one original species. Darwin envisioned this process as a branching event and diagrammed the process in the only illustration on the origin of species, Figure 18. 11a. Compare this illustration to the diagram of elephant evolution, Figure 18. 11b, which shows that as one species changes over time, it branches to form more than one new species, repeatedly, as long as the population survives or until the organism becomes extinct. Image A shows a sketch of lines branching into a tree shape. At the bottom are 11 vertical lines labeled A through L. These then are branched out as they move up across the page through 14 rows labeled with Roman numerals. Some branches make a straight line from the bottom row to the top row. Others keep branching out further at each row. And some are straight partway through the rows until they connect to an existing branch or form no connection and instead stop. The top 4 rows contain lines branching out from 6 locations on row 11 to 15 individual final designations on row 14. Illustration B shows the evolution of modern African and Asian elephants from a common ancestor, the Palaeomastodon. The Palaeomastodon was similar to modern elephants. However, it was smaller and had a long nose instead of a trunk. Side branches of the elephant evolutionary tree gave rise to mastodons and mammoths. The mammoth is more closely related to modern elephants than the mastodon. Figure 18. 11. The only illustration in Darwin's On the Origin of Species is A diagram showing speciation events leading to biological diversity. The diagram shows similarities to phylogenetic charts that today illustrate the relationships of species. B. Modern elephants evolved from the Palaeomastodon, a species that lived in Egypt 35–50 million years ago. For speciation to occur, 2 new populations must form from one original population, and they must evolve in such a way that it becomes impossible for individuals from the 2 new populations to interbreed. Biologists have proposed mechanisms by which this could occur that fall into 2 broad categories. Allopatric speciation, allo equals other, patric equals homeland, involves geographic separation of populations from a parent species and subsequent evolution. Sympatric speciation, sim equals same, patric equals homeland, involves speciation occurring within a parent species remaining in one location. Biologists think of speciation events as the splitting of one ancestral species into 2 descendant species. There is no reason why more than 2 species might not form at one time, except that it is less likely, and we can conceptualize multiple events as single splits occurring close in time. Allopatric speciation, a geographically continuous population, has a gene pool that is relatively homogeneous. Gene flow, the movement of alleles across a species range, is relatively free because individuals can move and then mate with individuals in their new location. Thus, an allele's frequency at one end of a distribution will be similar to the allele's frequency at the other end. When populations become geographically discontinuous, it prevents alleles' free flow. When that separation lasts for a period of time, the 2 populations are able to evolve along different trajectories. Thus, their allele frequencies at numerous genetic loci gradually become increasingly different as new alleles independently arise by mutation in each population. Typically, environmental conditions, such as climate, resources, predators, and competitors for the 2 populations, will differ, causing natural selection to favor divergent adaptations in each group. Isolation of populations leading to allopatric speciation can occur in a variety of ways. A river forming a new branch, erosion creating a new valley, a group of organisms traveling to a new location without the ability to return, or seeds floating over the ocean to an island. The nature of the geographic separation necessary to isolate populations depends entirely on the organism's biology and its potential for dispersal. If 2 flying insect populations took up residence in separate nearby valleys, chances are individuals from each population would fly back and forth, continuing gene flow. However, if a new lake divided 2 rodent populations, continued gene flow would be unlikely. Therefore, speciation would be more likely. Biologists group allopatric processes into 2 categories: dispersal and vicariance. Dispersal is when a few members of a species move to a new geographical area. And vicariance is when a natural situation arises to physically divide organisms. Scientists have documented numerous cases of allopatric speciation taking place. For example, along the west coast of the United States, two separate spotted owl subspecies exist. The northern spotted owl has genetic and phenotypic differences from its close relative, the Mexican spotted owl, which lives in the south, figure 1812. The northern spotted owl lives in the Pacific Northwest, and the Mexican spotted owl lives in Mexico and the southwestern portion of the United States. The two owls are similar in appearance, but with slightly different coloration. Figure 18:12. The northern spotted owl and the Mexican spotted owl inhabit geographically separate locations with different climates and ecosystems. The owl is an example of allopatric speciation. Credit Northern Spotted Owl. Modification of work by John and Karen Hollingsworth. Credit Mexican Spotted Owl. Modification of work by Bill Radke. Additionally, scientists have found that the further the distance between two groups that once were the same species, the more likely it is that speciation will occur. This seems logical because as the distance increases, the various environmental factors would likely have less in common than locations in close proximity. Consider the two owls. In the north, the climate is cooler than in the south. The types of organisms in each ecosystem differ, as do their behaviors and habits. Also, the hunting habits and prey choices of the southern owls vary from the northern owls. These variances can lead to evolved differences in the owls, and speciation likely will occur. Adaptive radiation. In some cases, a population of one species disperses throughout an area, and each finds a distinct niche or isolated habitat. Over time, the varied demands of their new lifestyles lead to multiple speciation events originating from a single species. We call this adaptive radiation because many adaptations evolve from a single point of origin, thus causing the species to radiate into several new ones. Island archipelagos like the Hawaiian Islands provide an ideal context for adaptive radiation events because water surrounds each island, which leads to geographical isolation for many organisms. The Hawaiian honeycreeper illustrates one example of adaptive radiation. From a single species, the founder species, numerous species have evolved, including the 6 in figure 1813. The illustration shows a wheel with the founder species at the hub. The spokes of the wheel are 6 modern honeycreeper bird species that evolved from the founder bird species. 5 of these birds eat insects and/or nectar and have long, thick beaks. These are the apapane, iwi, amakihi, akiapaleau, and Maui parrotbill. The Nihoa finch has a short, fat beak and eats insects, seeds, and bird eggs. Figure 18:13. The honeycreeper birds illustrate adaptive radiation. From one original species of bird, multiple others evolved, each with its own distinctive characteristics. Notice the differences in the species, beaks in figure 1813. Evolution in response to natural selection based on specific food sources in each new habitat led to evolution of a different beak suited to the specific food source. The seed-eating bird has a thicker, stronger beak, which is suited to break hard nuts. The nectar-eating birds have long beaks to dip into flowers to reach the nectar. The insect-eating birds have beaks like swords, appropriate for stabbing and impaling insects. Darwin's finches are another example of adaptive radiation in an archipelago. Link to learning. Watch this video to see how scientists use evidence to understand how birds evolved. Sympatric speciation. Can divergence occur if no physical barriers are in place to separate individuals who continue to live and reproduce in the same habitat? The answer is yes. We call the process of speciation within the same space sympatric. The prefix sim means same, so sympatric means same homeland in contrast to allopatric meaning other homeland. Scientists have proposed and studied many mechanisms. One form of sympatric speciation can begin with a serious chromosomal error during cell division. In a normal cell division event, chromosomes replicate, pair up, and then separate so that each new cell has the same number of chromosomes. However, sometimes the pairs separate, and the end cell product has extra sets of chromosomes in a condition that we call polyploidy, figure 18. 14. Visual connection. Aneuploidy results when chromosomes fail to separate correctly during meiosis. As a result, one gamete has one too many chromosomes, shown as n+1, and the other has one too few, shown as n-1. When the n+1 gamete fuses with a normal gamete, the resulting zygote has two n+1 chromosomes. When the n-1 gamete fuses with a normal gamete, the resulting zygote has two n-1 chromosomes. Figure 18. 14 aneuploidy results when the gametes have too many or too few chromosomes due to non-disjunction during meiosis. In this example, the resulting offspring will have two n+1 or two n1 chromosomes. Which is most likely to survive, offspring with two n+1 chromosomes or offspring with two n1 chromosomes? Polyploidy is a condition in which a cell or organism has an extra set, or sets, of chromosomes. Scientists have identified two main types of polyploidy that can lead to reproductive isolation of an individual in the polyploidy state. Reproductive isolation is the inability to interbreed. In some cases, a polyploid individual will have two or more complete sets of chromosomes from its own species in a condition that we call autopolyploidy. Figure 18. 15. The prefix auto means self, so the term means multiple chromosomes from one's own species. Polyploidy results from an error in meiosis in which all of the chromosomes move into one cell instead of separating. Autopolyploidy results in offspring with two sets of chromosomes. In the example shown, a diploid parent, 2n, produces polyploid offspring 4n. Figure 18. 15 autopolyploidy results when mitosis is not followed by cytokinesis. For example, if a plant species with 2n = 6 produces autopolyploid gametes that are also diploid, 2n = 6, when they should be n = 3, the gametes now have twice as many chromosomes as they should have. These new gametes will be incompatible with the normal gametes that this plant species produces. However, they could either self-pollinate or reproduce with other autopolyploid plants, with gametes having the same diploid number. In this way, sympatric speciation can occur quickly by forming offspring with 4n that we call a tetraploid. These individuals would immediately be able to reproduce only with those of this new kind and not those of the ancestral species. The other form of polyploidy occurs when individuals of two different species reproduce to form a viable offspring that we call an allopolyploid. The prefix allo means other, recall from allopatric. Therefore, an allopolyploid occurs when gametes from two different species combine. Figure 18. 16 illustrates one possible way an allopolyploid can form. Notice how it takes 2 generations, or 2 reproductive acts, before the viable fertile hybrid results. Alloploidy results from viable matings between two species with different numbers of chromosomes. In the example shown, species 1 has 2 pairs of chromosomes, and species 2 has 3 pairs of chromosomes. When an unreduced gamete from species 1, with 2 pairs of chromosomes, fuses with a normal gamete from species 2, with 3 unpaired chromosomes, a hybrid zygote with 7 chromosomes results. An offspring from this mating produces an unreduced gamete with 7 chromosomes. If this unreduced gamete fuses with a normal gamete from species 1, with 3 chromosomes, the resulting offspring will have 10 viable chromosomes, 5 pairs of chromosomes. Figure 18. 16 alloploidy results when two species mate to produce viable offspring. In this example, a normal gamete from one species fuses with a polyploidy gamete from another. 2 matings are necessary to produce viable offspring. The cultivated forms of wheat, cotton, and tobacco plants are all allopolyploids. Although polyploidy occurs occasionally in animals, it takes place most commonly in plants. Animals with any of the types of chromosomal aberrations that we describe here are unlikely to survive and produce normal offspring. Scientists have discovered more than half of all plant species studied relate back to a species evolved through polyploidy. With such a high rate of polyploidy in plants, some scientists hypothesize that this mechanism takes place more as an adaptation than as an error. Reproductive isolation. Given enough time, the genetic and phenotypic divergence between populations will affect characters that influence reproduction. If individuals of the two populations were brought together, mating would be less likely, but if mating occurred, offspring would be non-viable or infertile. Many types of diverging characters may affect the reproductive isolation, the ability to interbreed, of the two populations. Reproductive isolation can take place in a variety of ways. Scientists organize them into two groups: prezygotic barriers and postzygotic barriers. Recall that a zygote is a fertilized egg, the first cell of an organism's development that reproduces sexually. Therefore, a prezygotic barrier is a mechanism that blocks reproduction from taking place. This includes barriers that prevent fertilization when organisms attempt reproduction. A postzygotic barrier occurs after zygote formation. This includes organisms that don't survive the embryonic stage and those that are born sterile. Some types of prezygotic barriers prevent reproduction entirely. Many organisms only reproduce at certain times of the year, often just annually. Differences in breeding schedules, which we call temporal isolation, can act as a form of reproductive isolation. For example, two frog species inhabit the same area, but one reproduces from January to March, whereas the other reproduces from March to May, figure 1817. Photo A shows Rana aurora, a beige frog with green spots. Photo B shows Rana boylii, a brown frog. Figure 18:17. These two related frog species exhibit temporal reproductive isolation. A. Rana aurora breeds earlier in the year than B. Rana boylii. Credit: a modification of work by Mark R. Jennings, USFWS. Credit B. Modification of work by Alessandro Catanazzi. In some cases, populations of a species move, or are moved to a new habitat, and take up residence in a place that no longer overlaps with the same species' other populations. We call this situation-habitat isolation. Reproduction with the parent species ceases, and a new group exists that is now reproductively and genetically independent. For example, a cricket population that was divided after a flood could no longer interact with each other. Over time, natural selection forces, mutation, and genetic drift will likely result in the two groups diverging. Figure 18. Illustration A shows the black Gryllus pennsylvanicus cricket on sandy soil, and illustration B shows the beige Gryllus firmus cricket in grass. Figure 18. Speciation can occur when two populations occupy different habitats. The habitats need not be far apart. The cricket A, Gryllus pennsylvanicus, prefers sandy soil, and the cricket B, Gryllus firmus, prefers loamy soil. The two species can live in close proximity, but because of their different soil preferences, they became genetically isolated. Behavioral isolation occurs when the presence or absence of a specific behavior prevents reproduction. For example, male fireflies use specific light patterns to attract females. Various firefly species display their lights differently. If a male of one species tried to attract the female of another, she would not recognize the light pattern and would not mate with the male. Other prezygotic barriers work when differences in their gamete cells, eggs, and sperm prevent fertilization from taking place. We call this a gametic barrier. Similarly, in some cases, closely related organisms try to mate, but their reproductive structures simply do not fit together. For example, damselfly males of different species have differently shaped reproductive organs. If one species tries to mate with the female of another, their body parts simply do not fit together. Figure 1819. Illustrations show 4 different types of damselfly reproductive organs. Each organ has a hook, but the shape and length of the hook varies, as does the shape of the organ itself. Figure 1819. The shape of the male reproductive organ varies among male damselfly species and is only compatible with the female of that species. Reproductive organ incompatibility keeps the species reproductively isolated. In plants, certain structures aim to attract one type of pollinator, simultaneously prevent a different pollinator from accessing the pollen. The tunnel through which an animal must access nectar can vary widely in length and diameter, which prevents the plant from cross-pollinating with a different species. Figure 1820. Illustration A shows a bee collecting pollen from a bright purple foxglove flower. The bee's body fits inside the bell-like flower. Illustration B shows a hummingbird drinking nectar from a long tube like trumpet creeper flower. Figure 1820. Some flowers have evolved to attract certain pollinators. The wide foxglove flower is adapted for pollination by bees, while the B, long, tube-shaped trumpet creeper flower is adapted for pollination by hummingbirds. When fertilization takes place and a zygote forms, postzygotic barriers can prevent reproduction. Hybrid individuals in many cases cannot form normally in the womb and simply do not survive past the embryonic stages. We call this hybrid inviability because the hybrid organisms simply are not viable. In another postzygotic situation, reproduction leads to hybrid birth and growth that is sterile. Therefore, the organisms are unable to reproduce offspring of their own. We call this hybrid sterility. Habitat influence on speciation. Sympatric speciation may also take place in ways other than polyploidy. For example, consider a fish species that lives in a lake. As the population grows, competition for food increases. Under pressure to find food, suppose that a group of these fish had the genetic flexibility to discover and feed off another resource that other fish did not use. What if this new food source was located at a different depth of the lake? Over time, those feeding on the second food source would interact more with each other than the other fish. Therefore, they would breed together as well. Offspring of these fish would likely behave as their parents, feeding and living in the same area and keeping separate from the original population. If this group of fish continued to remain separate from the first population, eventually sympatric speciation might occur as more genetic differences accumulated between them. This scenario does play out in nature, as do others that lead to reproductive isolation. One such place is Lake Victoria in Africa, famous for its sympatric speciation of cichlid fish. Researchers have found hundreds of sympatric speciation events in these fish, which have not only happened in great number, but also over a short period of time. Figure 18.21 shows this type of speciation among a cichlid fish population in Nicaragua. In this locale, two types of cichlids live in the same geographic location, but have come to have different morphologies that allow them to eat various food sources. Illustrations show two species of cichlid fish which are similar in appearance except that one has thin lips and one has thick lips. Figure 18.21 cichlid fish from Lake Apoyac, Nicaragua, show evidence of sympatric speciation. Lake Apoyac, a crater lake, is 1,800 years old, but genetic evidence indicates that a single population of cichlid fish populated the lake only 100 years ago. Nevertheless, two populations with distinct morphologies and diets now exist in the lake, and scientists believe these populations may be in an early stage of speciation. Learning objectives. By the end of this section, you will be able to do the following. Describe pathways of species evolution in hybrid zones. Explain the two major theories on rates of speciation. Speciation occurs over a span of evolutionary time, so when a new species arises, there is a transition period during which the closely related species continue to interact. Reconnection. After speciation, two species may recombine or even continue interacting indefinitely. Individual organisms will mate with any nearby individual with whom they are capable of breeding. We call an area where two closely related species continue to interact and reproduce, forming hybrids a hybrid zone. Over time, the hybrid zone may change depending on the fitness of the hybrids and the reproductive barriers. Figure 18.22. If the hybrids are less fit than the parents, speciation reinforcement occurs, and the species continue to diverge until they can no longer mate and produce viable offspring. If reproductive barriers weaken, fusion occurs and the two species become one. Barriers remain the same if hybrids are fit and reproductive. Stability may occur and hybridization continues. Visual connection. Three different possible changes in the hybrid zone may occur over time. The first possible change, reinforcement, results when hybrids are less fit than either purebred species. Like a fork in the road, the species continue to diverge until hybridization no longer occurs. The second possible change, fusion, results when reproductive barriers weaken until two species become one. In this scenario, species initially diverge, but then join together. In the third scenario, stability, fit hybrids continue to be produced at a steady rate. Figure 18.22. After speciation has occurred, the two separate but closely related species may continue to produce offspring in an area called the hybrid zone. Reinforcement, fusion, or stability may result, depending on reproductive barriers and the relative fitness of the hybrids. If two species eat a different diet, but one of the food sources is eliminated, and both species are forced to eat the same foods, what change in the hybrid zone is most likely to occur? Hybrids can be either less fit than the parents, more fit, or about the same. Usually hybrids tend to be less fit. Therefore, such reproduction diminishes over time, nudging the two species to diverge further in a process we call reinforcement. Scientists use this term because the hybrid's low success reinforces the original speciation. If the hybrids are as fit or more fit than the parents, the two species may fuse back into one species, figure 18. 23. Scientists have also observed that sometimes two species will remain separate but also continue to interact to produce some individuals. Scientists classify this as stability because no real net change is taking place. Varying rates of speciation. Scientists around the world study speciation, documenting observations both of living organisms and those found in the fossil record. As their ideas take shape, and as research reveals new details about how life evolves, they develop models to help explain speciation rates. In terms of how quickly speciation occurs, we can observe two current patterns: gradual speciation model and punctuated equilibrium model. In the gradual speciation model, species diverge gradually over time in small steps. In the punctuated equilibrium model, a new species undergoes changes quickly from the parent species, and then remains largely unchanged for long periods of time afterward. Figure 1823. We call this early change model punctuated equilibrium because it begins with a punctuated or periodic change and then remains in balance afterward. While punctuated equilibrium suggests a faster tempo, it does not necessarily exclude gradualism. Visual connection. In the gradual speciation example, a founder species of bird diverges into one species with a hooked beak and another with straight beak. Over time, through gradual intermediate stages, the hooked beak gets longer and thinner, and the straight beak gets shorter and fatter. In the punctuated equilibrium example, as in the graduated speciation example, the founder species diverges into one species with a hooked break and another with a straight beak. However, in this case, the hooked and straight beaks remain unchanged for a significant period of time, and only then give rise to long, thin, and short, fat beaks. Figure 18. 23. In A, gradual speciation, species diverge at a slow, steady pace as traits change incrementally. In B, punctuated equilibrium, species diverge quickly and then remain unchanged for long periods of time. Which of the following statements is false? Punctuated equilibrium is most likely to occur in a small population that experiences a rapid change in its environment. Punctuated equilibrium is most likely to occur in a large population that lives in a stable climate. Gradual speciation is most likely to occur in species that live in a stable climate. Gradual speciation and punctuated equilibrium both result in the divergence of species. The primary influencing factor on changes in speciation rate is environmental conditions. Under some conditions, selection occurs quickly or radically. Consider a species of snails that had been living with the same basic form for many thousands of years. Layers of their fossils would appear similar for a long time. When a change in the environment takes place, such as a drop in the water level, a small number of organisms are separated from the rest in a brief period of time, essentially forming one large and one tiny population. The tiny population faces new environmental conditions. Because its gene pool quickly became so small, any variation that surfaces and that aids in surviving the new conditions becomes the predominant form. Link to learning. Visit this website to continue the speciation story of the snails.